The field you're asking about is epigenetics. It's the study of how environmental and lifestyle factors can cause heritable changes in gene expression without altering the underlying DNA sequence. Think of it as a set of instructions that tells your genes when to turn on and off. While your DNA is the hardware, epigenetics is the software that dictates how the hardware operates. Epigenetic changes are like switches or dials on your genes. These switches can be turned on or off by various factors, leading to changes in how your body functions. The primary mechanisms of epigenetics include DNA methylation. This is the most studied epigenetic mechanism. A chemical group, a methyl group, CH3, is added to the DNA molecule, usually at a specific nucleotide called cytosine. When this happens in a gene's promoter region, the area that controls gene expression, it often acts like a brake, silencing the gene and preventing it from being expressed. Histone modification is a key epigenetic mechanism that regulates gene expression by altering the structure of chromatin. Histones are proteins that DNA wraps around to form nucleosomes, the basic unit of chromatin. Post-translational modifications to the histone tails can affect how tightly the DNA is wound, thereby controlling the accessibility of genes to the transcriptional machinery. Types of histone modifications. There are several types of histone modifications, each with a different effect. Acetylation. The addition of acetyl groups to lysine residues on histone tails, catalyzed by histone acetyltransferases, HATs, neutralizes their positive charge. This reduces the electrostatic attraction between the histones and the negatively charged DNA, causing the chromatin structure to loosen. This open configuration, known as euchromatin, makes the DNA more accessible to transcription factors and RNA polymerase, promoting gene expression. The removal of acetyl groups by histone deacetylases, HDACs, has the opposite effect, leading to a more condensed, transcriptionally inactive state called heterochromatin. Methylation. The addition of methyl groups to lysine or arginine residues is a more complex modification. Depending on the specific site and the number of methyl groups added, methylation can either activate or repress gene expression. For example, trimethylation of histone H3 at lysine 4, H3K4Me3, is generally associated with active gene promoters, while trimethylation at lysine 9, H3K9Me3, is typically linked to gene repression. Phosphorylation. The addition of phosphate groups can also affect chromatin structure and gene expression. Phosphorylation of histones is often a rapid response to cellular signals, and can work in conjunction with other modifications to regulate processes like mitosis and DNA repair. Ubiquitination. The attachment of a small protein called ubiquitin to histones can also influence gene expression, either by changing chromatin conformation or by recruiting other regulatory proteins. Non-coding RNA, ncRNA. Regulation non-coding RNA, ncRNA molecules play a crucial role in regulating gene expression at various levels, including transcriptional, post-transcriptional, and translational. They do not code for proteins but instead perform diverse functions by interacting with DNA, other RNA molecules, and proteins. Transcriptional regulation. Some ncRNAs can bind directly to specific DNA sequences to either block or promote the binding of transcription factors, thereby controlling gene activity. They can also act as guides, recruiting chromatin-modifying enzymes such as histone methyltransferases to specific gene loci to alter the epigenetic landscape. Post-transcriptional regulation. A well-known example of this is the action of microRNAs, miRNAs. These small ncRNAs bind to complementary sequences on messenger RNA, mRNA molecules, leading to the degradation of the mRNA or the inhibition of its translation into a protein. Other ncRNAs can act as sponges, soaking up miRNAs and preventing them from targeting their intended mRNAs. Chromatin remodeling. Long non-coding RNAs, lncRNAs, can interact with chromatin remodeling complexes to affect the structural state of chromatin, which in turn influences gene transcription. For instance, some lncRNAs can help to form or maintain a condensed chromatin structure leading to gene silencing. Non-coding RNA, ncRNA. This video provides a general overview of the role of non-coding RNA in cellular processes, including gene regulation.